Our next presenting company is AsoThera, which is developing new therapies for orphan diseases based on technology developed at Coastal Harbor Labs and already proven by Big Pharma. Presenting this morning for AsoThera is Isabel Aznares.
Thanks, Neil, for the introduction, and thanks for giving us the opportunity to present AsoThera to you. AsoThera's mission is to develop antisense drugs with our proven platform to treat orphan diseases, currently a $100 billion market. Our technology is based on innovative research from the Craner Lab at Coastal Harbor Laboratory, which has been funded by more than $2 million in NIH and foundation money. In collaboration with ICES Pharmaceuticals, we have developed a drug using our platform to treat a disease called spinal muscular atrophy. And here I'm going to show you two videos. The first video shows two mice. One is very small and fragile, and that mouse has spinal muscular atrophy. These mice don't live past day 10. On the right-hand side, there are two videos. There's another video with 4 mice. 2 of those mice have spinal muscular atrophy, and 2 are healthy. The 2 mice with spinal muscular atrophy were treated with our drug, and now you can barely distinguish them from the healthy mice. We have extended their life expectancy by 26 times, and now these mice live a year of age. This drug is currently in phase 2 clinical trials and is carried out by ICES in partnership with BioTech. And this is how our platform works. From genes to protein, there's a messenger that brings the message coded in the genes to the protein-making machinery. And that messenger is called RNA, it's a messenger RNA. And we know that this messenger RNA is under positive and negative pressure carried out by activator proteins and repressor proteins. And the balance of these proteins makes sure that this messenger is processed correctly so that proper protein production is achieved. In the context of a disease mutation, that messenger is handicapped already. So we learned that having that additional negative pressure carried out by a repressor is redundant. It's like hitting that messenger twice. And that leads to no protein production, which is the vast majority of the cause of the vast majority of genetic diseases. So by using antisense oligonucleotides, which are DNA or RNA synthetic short molecules, we can direct them specifically to the regions where those repressors bind, thereby blocking the access of the repressor and restoring or overcoming the effect of the mutation and leading to back to proper protein production. So using this platform and our expertise, we address a huge unmet medical need which are orphan genetic diseases. These diseases affect more than 300 million individuals worldwide. And despite the high unmet medical need, only 5% of these diseases have approved treatment. Currently, these treatments only manage symptoms with little impact on life expectancy. There's a huge economic power to orphan drugs, and the reason for this economic power are the research and development related drivers and economic and commercial drivers, and such as tax credits, FDA fees are waived, greater regulatory success, smaller clinical trials, and the commercial side, 7-year market exclusivity, premium pricing, favorable reimbursement. And because of all these drivers, orphan drugs' expected return on investment is 1.7 times that of non-orphan drugs. And it is estimated that by 2018, the orphan drug market is going to be $127 billion. With our platform, we're capable of treating many of these genetic diseases. Currently, we have 3 indications in our pipeline. Cystic fibrosis is the most common genetic disease in the Caucasian population. It has a life expectancy of approximately 38 years of age, and with very costly treatment that manages quality of life. Familial dysautonomia is an ultra-rare genetic disease that has a life expectancy of 30 years of age. And glioblastoma is the most common brain tumor with a life expectancy of 4.5 months without treatment and only up to a year and a half with treatment. So there's a large medical need for these diseases. We're not alone in the world of antisense technology or orphan disease drug development. However, our company is the only one capable of increasing protein expression, which is what's needed in genetic diseases, with high specificity because we target the gene of interest and easy delivery. This is the general timeline if we were to start a drug right now to IND. And in the best case scenario, it would take us approximately 2.5 years to get to IND. And here are the variables and the resources needed to achieve that, at an estimated cost of $1 million. Right now, with each of the indications we have in the pipeline, this is where we're at, and we are seeking approximately $2.2 million to get these drugs to in vivo proof of concept, and the overall goal of the company is to get these drugs to IND. And once we are at IND, the plan is to license each one of these targets to a larger pharmaceutical company and partner with them throughout clinical trials. Here are examples of licensing agreements, recent licensing agreements of drugs, antisense drugs that are comparable to ours. So we expect our numbers to be similar to these ones. Once the drug is in the market, here's the top 10 orphan drugs in the market currently. As you can see in this, oops, sorry about that. In this column, you see the pricing of these drugs per patient per year, and this is the revenue in the year 2012 for each of these drugs. So to summarize, we offer validated technology with favorable regulatory strategy given that we're treating orphan genetic diseases, an attractive revenue model based on the Genzyme orphan disease model, and high level of reimbursement given the highly unmet medical need. This is our team, Adrian Craner and I are the scientific founders. We're a spin-out of Cold Spring Harbor Laboratory, and we're currently seeking for a CEO. Thank you.
Questions?
I'm not sure how to articulate this, but in the, I probably asked this before, but in the first technology slide, you talk about a complementary strand coming in and blocking. But it looks sort of serial, right? If you get there first, it blocks. But my guess is it's not really serial. So it's just that enough complementary strands are coming in and binding and protecting that you get the impact that you get.
Yeah,
is that correct?
It will compete, of course. Yeah.
Can you say a couple words about your IP position currently?
Yes. So we're currently negotiating an option to license agreement with Cold Spring Harbor Laboratory. Oh, and sorry, and we have provisional patents filed for each of these applications.
Good. Thank you.